So, homologous structures are one of the best sources of information to construct these phylogenetic relationships. The example here I have on the right with the skull and the teeth. Often fossils, one of the important structural features that they use are the number and arrangement. Sorry, where's my cursor? Here we go. The number and the arrangement of the teeth, something that's called dentition, but of the teeth, is important in establishing relationships between fossil species and establishing these phylogenetic trees, these evolutionary family trees of organisms. So let's look at a process that's called convergent evolution. This is where superficially similar structures like wings, that's a wing, and that's a wing on a bird, a bat, and an insect. So they're all wings. So superficially they might appear similar, but they are from different evolutionary branches, and the result of the similarity is through natural selective pressures that have caused analogous adaptations. So similar adaptations, but not homologous structures, because these organisms are not closely related. This is a bird, a bat's a mammal, and a butterfly is an insect. So the cause of the somewhat similar structures of these wings is due to convergent evolution, not similar evolutionary relationships. Okay? So the similarity is due to convergence. Due to convergence is referred to as an analogy. These are not homologous characteristics or homologous structures. If you remember, a homologous structure or characteristic is a similar structure due to a similar evolutionary background or relationship. Okay? So these are analogous structures or analogous characters. For example, these two things down here might look like cacti or cactuses, and in reality the one on the left is a cactus and the one on the right is not. It's something called a euphorbs. These are very common in like Africa, and these are very common in the Americas. The reason they appear similar, they have similar superficial structures or morphology, is because of convergent evolution. So they are analogous in terms of their structures. They are not homologous structures. These are actually not closely related. If we look at a shark, a dolphin, and a puma, the dolphin is actually much more closely related to the puma than it is to a shark. Dolphins and pumas are both mammals. A shark is a fish. So they are both all vertebrates, but their evolutionary relationships, a dolphin and a shark, are very distant. The reason they're both very streamlined in their shapes and have fins and tail fins is due to convergent evolution. Their structural shapes, fins and tail fins, and streamlined body are analogous characters or structures, not homologous. These two are not closely related. But the reason their shapes are somewhat similar is due to convergent evolution, right? They have analogous shapes, or their structures are analogous structures or analogous characters. Okay, so we talked about comparing embryological development of two species to reveal if homology is apparent or not in the adult. A chicken and a human were both vertebrates. We have certain similar homologous structures, especially embryologic structures, and some are lost in the adult and become other things. But these homologous structures, again, homologous, meaning a relatively close relationship, or a closer relationship than, say, with other organisms. Okay? And again, we talked about comparing DNA sequences to help clarify evolutionary relationships. If we look at the amino acids and their sequence in that hemoglobin protein or polypeptide, humans and rhesus monkeys have a very similar match. If you compare a rhesus monkey to the human, So there's a 95% match with the hemoglobin polypeptide, meaning the hemoglobin gene is similar in both of these. Much more similar than a human and, say, a chicken or a human and a frog. There's only a 54% similarity. So the greater the similarity in DNA sequences, which result again in polypeptide sequences of amino acids, means the closer the evolutionary relationship, the greater the dissimilarity, or the lower the similarity, the more distant the evolutionary relationship. And this is that figure we saw in a previous chapter. So the closer the evolutionary relationship, the more similar the DNA sequences should be, and that's been found many, many times when biologists look at the DNA sequences, the genetics of different organisms. And these DNA sequences, this genetic evidence, along with embryological evidence and homologous characters, that evidence, this is all used to establish relationships, phylogenies. So let's move into classification schemes. So cladistics is a branch of biology, systematics, or taxonomy where organisms are grouped by common ancestry to form what's called the clade. So this is a grouping of ancestral species and all its evolutionary descendants that form a distinct branch in an evolutionary tree or in a phylogeny. So if you look at the figure here, the way cladistics often works is they compare an in-group of the organisms that the biologists or scientists are interested in examining with an out-group, something that's distantly related. Okay? So the in-group is a group of organisms that is actually being analyzed. What we see here is actually a clade. It's a very simple one, but it's a clade that shows the common ancestor is a mammal with hair or fur and mammary glands. So that leads to things like a duck-billed platypus, or a kangaroo, and or a beaver. These are all mammals. Okay? The outgroup, in comparison to these guys, is an iguana, which is actually a reptile. Okay? So what we have here is a clade with the common ancestor here. And this is how cladistics normally works. So cladistics can clarify evolutionary relationships that weren't always apparent in other taxonomic classifications. So what we see here, this figure is an older way to represent relationships between reptiles, birds, and mammals. The orders Reptilia, Aves, and Mammalia. In reality, birds should belong into the Reptilia group, but this is an older classification scheme, which is actually incorrect. So biologists traditionally have placed birds and reptiles in separate classes of vertebrates. We now know that birds are more closely related to some reptiles versus others, and actually things like turtles are more distantly related to things like crocodiles and birds than they are to some of the other reptiles. So shared characteristics do indicate that birds and crocodiles make up one clade, simplified here, or crocodiles here and birds here. This whole grouping is a clade because there's their common ancestor, or that's where a common ancestor would be. And snakes and lizards make up an outgroup in this example. Okay? So what this is showing you here, crocodiles to birds, is from birds to crocodiles here. And then lizards and snakes are a more distant group, and even more distantly related are turtles and tortoises. So going back to examine the ancestor of the crocodiles, that is also inclusive with the ancestor, or including the ancestor of the lizards and snakes. Now we actually have and can form a larger clade, which includes their common ancestor. So if we look at that situation on the phylogeny on the left here, lizards and snakes would be here. So from this point, this common ancestor to all these groups, that would be a clade. If we go back to the common ancestor of all these and turtles, that's a clade there. If we go all the way back, we can actually go back and identify a common ancestor to the mammals and the reptiles and have even a larger clade. So the clade will be as big or as small as the investigator wants. The important thing about a clade is it has to include the common ancestor and all their known descendants. So if we eliminate the birds and say, well, those aren't reptiles, and look at it this way, then the reptilia group is not a true clade because it doesn't include this relatively closely related group of the birds. So birds are actually reptiles. They're feathered, warm-blooded reptiles, but they actually are reptiles. Let me see. Okay, so phylogenetic trees are hypotheses about evolutionary history. Linnaeus placed all forms of life into two groups only. They were either animals or plants. A two-kingdom system prevailed for about 200 years. With more advancement in science and in technology, it got a little more complicated, but still incorrect. By the mid-1900s, a five-kingdom system was proposed, with all the prokaryotes in one group, bacteria, for example, in one big group, a kingdom, and then the eukaryotes into four other kingdoms, protista, protists, plants, animals, and fungi. And this was the dominant classification system for quite a long time. More advancement, late 1900s, with molecular studies and cladistics work, led to what's called the three-domain system, with two domains of prokaryotes, bacteria and archaea, something we talked about a ways back now, and then one domain of the eukaryotic organisms, those with true nucleated cells. The domain eukarya with its kingdoms plantae, fungi, animalia, and protista, which is actually a group that still needs lots of taxonomic work to clarify it. So earliest organisms, domain bacteria, more recently appearing evolved archaea, and then the eukarya. Okay, so that's what we have now, three-domain system with lots of work clarification to do, especially on the protists. Okay, so a little video here on classification schemes. This thing is only a couple minutes long. We'll squeeze it in here.
In the early days of biology, all organisms were classified as either plants or animals. Single-celled eukaryotic organisms such as algae and paramecium were assigned to one of the kingdoms according to whether they are photosynthetic or not. Fungi and prokaryotes were grouped with plants because fungi are sedentary and prokaryotes have cell walls. A five-kingdom scheme was devised because some organisms don't fit well into a two-kingdom system of classification. The simple cells of prokaryotes set them apart in their own kingdom, Monera. Organisms in the other four kingdoms all have eukaryotic cells. Fungi are not photosynthetic, so they are given their own kingdom. The kingdom Protista is an assortment of eukaryotes that don't fit into the other kingdoms, mostly unicellular organisms such as paramecium, unicellular algae, and multicellular algae. The five-kingdom scheme divides living things into two fundamentally different groups, prokaryotes and eukaryotes. New data suggests that two kinds of prokaryotes diverged early in the history of life, and thus there are actually three major categories of organisms. A new classification system assigns more significance to the ancient split by creating a level of classification larger than a kingdom, a new category called a domain. The two domains of prokaryotes are bacteria and archaea. The domain eukarya encompasses all of the kingdoms of eukaryotes. Taxonomists are in the process of sorting out various kingdoms of prokaryotes within the domains bacteria and archaea, and also splitting the protists into several kingdoms within the domain eukarya.
All right. So we'll finish up this last part in the last lecture here for this chapter, which won't be very long.